Yeah, I'm just going to be talking about a bunch of different projects that have been going on at Kew, looking into the genetic basis of resistance to ash dieback. And I'll start with the sort of acknowledgement. So there's many different projects I'm going to be talking about, all of which have had a lot of different partners involved, and there's particularly the majority of funding coming from Defra, the CFP, and UKRI via NERC, and our many partners. And there are also lots of people, there's Richard in there as well, involved in all these projects. There are sort of too many to list, and I worry I might miss someone off. So just to thank everyone, and in particular I should probably just mention that Laura Kelly and Richard Buggs at Kew have led most of the work on the projects I'm going to be talking about. So I don't need to introduce ash dieback too much. Ash is obviously an important tree in our treescapes and is suffering a lot from ash dieback. It's a big problem. The good news is that there are, as Joe was mentioning, some trees that seem to be showing high levels of resistance to the disease. And a really key thing about that resistance is that it's partially heritable. And so that means that your resistant trees are more likely to produce resistant offspring than susceptible trees. But there's also an environmental component to it. So, it might be that a resistant tree still, if it grows in a particularly poor environment, might end up getting highly infected by ash dieback and vice versa. So there are these two different components to it. And we've been interested in identifying the genetic basis of that genetic component of resistance. And so what do I mean by that? Well, you've got your resistant and your susceptible tree there, and each of them have got their genome, which consists of millions and millions of base pairs of DNA. And most of that DNA is just the same between individuals, but there are also millions of differences, potentially, between any two individuals. And not all of these are going to be involved in ash dieback resistance. Most of them might not have any effect on any traits at all, but there might be some that seem to have an effect on it. So you might find that if, lots of trees that have a C in that particular position, the fifth base pair of this very small genome are more susceptible, and the ones that have a T in that position are more resistant, then that is giving you an idea of the genetic basis of the trait. And in many cases, there's not going to be just one single nucleotide conferring resistance, but that's the sort of thing we're looking into. And why should anyone care about this? So one of the reasons to try and identify the genetic basis of resistance is that it can allow you to select the best trees for something like a breeding program. So as I was saying, you might appear to be resistant, and that might be based on having, genetic variants associated with resistance. But you might also have just been lucky growing in a nice place. And so it might be that you then select those trees, and then when you grow them up and produce progeny, actually they don't produce very resistant progeny, and vice versa. Whereas if we know the genetic variants associated with resistance, we can screen trees before committing to growing them up in a trial. And because your genetic code is conserved throughout your life, you can do this on very small trees, genotype them, and then carry on with the ones that are the best, rather than wasting lots of time and space on all of your trees. You can also use this sort of information to try and select better crosses. So particularly if you've got multiple different genetic variants associated with resistance, you can try and combine them together to produce more resistance than you might have had in the parents. So on the, I guess that's on the left, if you've just got the same genetic variant in your two parents, the offspring will inherit that, and it might be about as resistant as the parents were, which is fine. But if you have two trees that are resistant, but there's a different sort of underlying genetic basis to each of their resistance, then some of their offspring might have more resistance variants in them than either of the parents, and therefore you might get an even more resistant tree than we currently see, which would be nice. You can also go out into natural populations and screen for variants associated with resistance to give you an idea of, which woodlands might be particularly susceptible or not, or have the potential to eventually produce some resistant offspring. And speaking of offspring, we can use this sort of data to test for things like natural selection. So if you were seeing a big increase in the number of these resistance variants in new generations of ash, that might tell you that natural selection is happening, and that could give you an idea of how quickly populations could evolve resistance to ash dieback. So then, what has research at Kew uncovered? There's quite a few things to sort of rattle through. So Firstly, we took advantage of these FR field trials that Joe was mentioning in 2017, and did a lot of DNA sequencing of the unhealthy individuals and healthy individuals, and looked for genetic variants that seemed to be associated with being healthy. And that identified thousands of these single base pair variants associated with ash dieback resistance. So it's not just a single base pair or a couple of loci, it seems to be lots and lots of very small effect loci. And you could use these to try and classify new trees outside that initial data set and see whether you could sort of use the information to identify whether or not they were healthy or sick just based on their genetics. And that seemed to work about 2/3 of the time to classify them as healthy versus unhealthy. But an important thing to note is that the top trees in terms of their genetics were almost always in the healthy category. So you could use it to get a pretty good idea of which your best trees might be to go forward with. And the sort of take-home message from this is, yes, that there's not a single resistance gene, but there are lots of them, and also that genomic data can be used to identify these high-performing trees. And that was published in this paper. And then we went out, well, I wasn't involved in this study, but we, Q, as an organisation, went out into a natural woodland in 2019, and then did a lot of sequencing of both adult trees that were established pre-pandemic and the offspring of them that were established post-pandemic. And we're looking for shifts in frequency in these resistance variants that were identified in the previous study. And what was found there is that these resistant variants were more frequent in the younger populations than would have been expected if there was no natural selection happening. And they estimated that something like 30% of the saplings that will have inherited the fewest resistance variants will have died, leaving the rest of them being more resistant than the ones before. And the increase in the frequency of a variant was correlated with how big an effect it was predicted to have in the trials. And so that suggests that natural selection for resistance is occurring, at least in this one wild population, and that the next generation might be more resistant to the disease than the previous generations, and it's a sort of nice further validation that these variants are, involved in resistance, not just in the field trial setting, but in a wider series of settings. It's worth just noting that this younger generation is going to be more resistant than the previous generation, but it's not necessarily going to be completely resistant to the disease, and it's not really known, how many of those saplings might actually survive to reproductive maturity. So it's, a step towards resistance, but how much more would be needed, I think, is a little unknown still. And then to just quickly mention a few of the projects that are ongoing, those are the ones that have been published. We're identifying more types of genetic variants. So the previous studies are just based on these single nucleotide changes between individuals, but there are also other genetic variants like big insertions or deletions of sequence, and those can be particularly important in things like disease resistance, but are a bit hard to characterize, and we've produced this ash pangenome that allows us to better do that in future studies going forward. There's also a CFP project that started last year that is going back to the FR field trials and resampling the surviving trees, and that allows us to look at a sort of longer term survival, and we can compare individuals that were sampled in 2017 and have subsequently died to those that have survived, and so that allows you to get an idea of the longer term survival. There are also a few projects going on looking at whether ash dieback disease stress is actually accelerating the evolutionary process in some ways. So the stress of being affected by disease can potentially increase things like your mutation rate or your recombination rate and the formation of structural variants. And so we're sort of looking into how that might be involved in the sort of evolutionary story of how this will go forward. And there are a few projects going on at the moment that are sort of directly relating to how one might do a breeding program and accelerating it. So one thing with ash is that it has these two self-incompatibility groups, and so you can only cross trees from one group to the other, you can't cross them within a group, and we can sort of identify using genetics which trees belong to which group without having to wait for them to grow up to flower and find out. We're also doing some simulation-based work, looking at what the best sort of crosses might be, using our knowledge of resistance variants, and also a recombination map for ash, which has been developed. And there is also a project that we're collaborating with NIAB and the University of Birmingham to try and induce rapid flowering in ash, because of course one of the bottlenecks for doing tree breeding is how long it takes to go from a seed to a flowering individual. And so with high temperature and, or well, I'm not sure it's high temperature, but changes in temperature and CO2 levels can potentially induce rapid flowering, which could accelerate a breeding program. So to just, the key takeaways from what is published so far is that we've got this partially inherited resistance to ash dieback, and that seems to be underpinned by thousands of variants with small effects, and these can be used to predict which trees are healthy, at least in field trials, and you can take that information out into wild populations and find that they are indeed shifting as well, consistent with natural selection going on in the wild. And I'm a bit short on time, so there is all sorts of exciting ongoing research as well. We're looking to sort of deepen our understanding of the genetic basis of resistance and try and think about how we can best apply that to something like a breeding program for ash. So yeah, thanks very much. That's it from me.